And welcome back. So we're going to get right in and finish up our innate defense mechanisms. So we're still in that second line of defense, and we're going to talk about fever. So fever is higher than normal body temperature. So normal body temperature is about 37 degrees Celsius or 98.6 degrees Fahrenheit. Anything above that would be considered a fever. The temperature center of the body is the hypothalamus. And so what a fever does is it resets the temperature to a higher degree. And this is usually associated with chemicals that are released in our system that can cause the hypothalamus to reset at a higher temperature. So the function of a fever is to inhibit certain materials from being released that are needed by bacteria. It also inhibits bacterial replication. So not only because they don't have the proper cofactors, but also because the bacteria grow best at certain body temperatures. So if your body temperature has increased, it can actually inhibit growth for that reason. Fever also increases repair mechanisms. So anytime you increase the temperature slightly, it's going to cause um, an increase in your metabolic reaction rate. Hello. And when you have increased metabolism, that means you're going to increase repairing tissues and protecting the body. So what are some benefits of fever? It inhibits reproduction of bacteria and viruses. It promotes interferon activity, increases activity of adaptive immunity, and accelerates tissue repair. So here is a fever, or here's a germ that's on Motrin, which is keeping your body temperature where you want it to be for normal if you're not ill. This bacteria can then replicate relatively easily. There's no inhibition of iron being released. Over here we have a fever, there's a 102. The germ can't replicate effectively. It's not going to work as effectively. So certain medicines that reduce body temperature, though they make you feel better, they're actually not benefiting you. They're actually causing more problems than they are causing benefits. That being said, if you have too high of a fever, that can actually lead to tissue damage as well. So any fever that's above like 105 is a serious issue that needs to be taken care of by the hospital. So if your body temperature goes above 105, you're at risk for cell damage, like nerve damage, brain damage in that case. And so you do need to be seen by a doctor or by the ER so that they can fix that problem. So I probably should have used those last two slides on the last video, but I didn't, so apologies. You should now be able to answer the following questions. What does innate mean? How does innate differ from adaptive? What does immunity mean? What are the two lines of defense under innate? What is the line of defense under adaptive? What are the mechanisms of the first line of defense? What are the mechanisms of the second line of defense? And now we're going to get into the adaptive mechanism. So now we're looking at immunity. The adaptive defense recognizes and acts against a specific invader. So it's going to be very antigen specific. We have white blood cells that only recognize one pathogen, and those are our B&T lymphocytes. The immunity is systemic, so it's going to move throughout our body, moves through the lymphatic tissue as well as through our blood to check for any potential pathogens. And we produce memory. Memory, our adaptive immune system, has cells that are called memory cells. Memory cells remember the pathogen, and then they can mount an attack immediately upon encountering that pathogen a second time. So it takes time for the immune system to become activated and to mount an army of defense, which is why when you do get sick, you usually are sick for a week or two. And then the immune system has mounted its or has produced its army and it attacks the pathogen, and then you get better. But then you don't get that illness again because you have those memory cells, and those memory cells are going to remember the pathogen and mount an attack so fast that the second time you get infected with the same pathogen, the immune system will take out that pathogen super fast. So there's two types of immunity. We have humoral immunity, which is associated with B lymphocytes. B lymphocytes produce antibodies, so it's also known as antibody-mediated immunity. And it is immunity that attacks the pathogen directly. And then we have cellular immunity. This is your T lymphocytes, and they attack body cells that are infected. So it's also known as cell-mediated immunity. So whereas your B lymphocytes produce antibodies that attack a pathogen directly, these T lymphocytes attack cells of the body that are infected with pathogens. So what is an antigen? An antigen is a substance that is capable of causing your immune system to respond. So there are lots of types of antigens. The most common antigens that we think about when we're talking about antigens are proteins, nucleic acids, carbohydrates, lipids, pollen grains, microbes. These are all called non-self antigens. So then we also have self-antigens. Self-antigens are antigens that are found on the surface of our own cells. So they're basically proteins that say, hey, I am me. I am who I am. And self-antigens don't trigger an immune response in us, but they could trigger an immune response in someone else, which is why we can't just donate blood or bone marrow or organs to just anyone, because my cells are okay in my body, but my cells could cause a lot of problems in someone else's body. A hapten is an incomplete antigen. And so haptens are things like poison ivy is an example of a hapten. And it has to link up with a protein inside of our body. And once it does, then it can cause an immune response. So here's an example of a hapten. Here is a self protein. This could be like albumin. Apart, neither is going to cause us problems. But when they link up, they can become a deadly pathogen or antigen. So at this point, you should be able to tell me the three aspects of the adaptive defense mechanism. What are the two types of immunity? What is an antigen? What is the difference between a self and a foreign antigen? And what is a hapten? We're going to continue on. I'm going to talk about the different types of lymphocytes and antigen-presenting cells. So lymphocytes are the cells that produce, so certain types of lymphocytes, I should say, are the cells that produce your adaptive immunity. We have B lymphocytes that produce antibodies, and they function in humoral immunity. Then we have T lymphocytes, which are your cells that produce cell-mediated immunity. Antigen-presenting cells are cells that can phagocytize specific antigens or different types of antigens. They're not specific, just any antigen. And then they can present those antigens to the body or to our immune system. So where do lymphocytes come from? Lymphocytes are white blood cells, and so they're going to arise from the same stem cell that all of our other blood comes from, a hemocytoblast. And all blood is produced in bone marrow. So we're producing our blood in the bone marrow, including our B and T lymphocytes, but where the lymphocyte matures is going to depend on if it is going to become a B cell or a T cell. So it's going to maturate in the thymus, then it becomes a T cell. If it maturates in the bone marrow, it becomes a B cell. And so I always like this picture, bone marrow prep school. So here you have where bone marrow is where all the cells are produced, and then T cells leave and they move to the thymus where they're going to become immunocompetent. B cells stay here and they become immunocompetent right here in the prep school. So immunocompetence is the capability to respond to a specific antigen that appear on the lymphocyte surface. But you're also not just going to become immunocompetent, but you're also going to recognize the difference between self and non-self. So they identify the foreign antigens and they differentiate foreign from self antigens. If they cannot do that, then they are destroyed. So they become self-tolerant as well as being able to identify foreign antigens. B cells do the same thing in the bone marrow. So once they become immunocompetent and are self-tolerant, then they can move to different lymphatic tissues, maybe lymph nodes, maybe the spleen, maybe the Peyer's patches, and they wait. So at this point, they are considered naive cells. Naive cells are cells that have not come in contact with a pathogen yet. So they will become activated when they come in contact with that pathogen and they get stimulated. So here we have, oh wait, okay, let's do this. Here we have lymphocytes in the bone marrow that are produced by that hemocytoblast. T lymphocytes leave the bone marrow and they migrate to the thymus. B lymphocytes stay in the bone marrow. And they're going to develop immunocompetence in the bone marrow, whereas T lymphocytes become immunocompetent in the thymus. Once that happens, then they leave. They move into the spleen or lymph nodes, and they wait. They're considered naive at this point, but they are immunocompetent. So they're just waiting to encounter an antigen, and once they do, they will become active. So once an antigen activates them, the immunocompetent cell then will immediately start to differentiate into different types of cells and proliferate, producing multiple copies of that cell. So antigen presenting cells are cells that produce certain or that phagocytize materials, and then once they have phagocytized the antigen, instead of releasing it via exocytosis, they take a piece of that protein and they incorporate it into one of their own proteins and then put it at the surface of the cell. Basically, this says, hey, I am an infected cell, and now, or I have, some type of a pathogen is going around in my body. So now you can look at this and you can become activated. So there are three types of antigen presenting cells: dendritic cells, which are found in your integumentary system, macrophages found everywhere, and B lymphocytes. So we'll look at humoral immunity, but hold on one second. I'm going to finish this up, so it'll be a little bit long, but I only have about 4 slides left, and then I'll start the next video. So B lymphocytes function in your humoral immunity. And what they do is when they bind to an antigen, that binding activates the lymphocyte, and the lymphocyte immediately will start producing clones of itself. And so these clones have the same protein receptors. And then these clones become plasma cells. Plasma cells are the cells that produce antibodies, and they basically release tons and tons of antibodies. These antibody producing cells only last for about 4 to 5 days. They expend a ton of energy, or a lot of energy, producing the antibodies, and then they die. But we do have a few of those B cells that become memory cells. So we proliferate and differentiate into either plasma cells or memory B cells. Memory cells will live a long time, and they will remember the pathogen. And this is what we call immunological memory. And so here we have that primary response. A pathogen binds. This immediately activates the B cell, and it starts to proliferate. It forms lots and lots of clones of itself, and then these clones will differentiate into either plasma cells or memory cells. Memory cells remember, whereas the plasma cells produce antibodies. And so if you're looking at a primary response to a pathogen versus a secondary response to a pathogen, if you were, in this case, injected with an antigen, so some pathogen got into your system, it would take your body about a week before your immune system actually starts to build up an army. So by about the beginning of the second week, you're going to have a high number of antibodies in your system that are going to fight off the infection. And so then you're going to start reducing the pathogen in your system until you have no pathogen in your system. And now you still have a few antibodies, though. The second time you get infected with the pathogen, immediately, not even a day goes by, and you already have an even higher number of antibodies that are going to fight off the infection before you can even show any type of discomfort to the disease. So you haven't even showed any symptoms of the disease. Your body's already fighting off the infection. And this is due to those memory cells. And so memory cells are what remember the pathogen, and I just thought this was the cutest little picture. Here, this is a memory T. We were talking about memory B cells, but memory B and memory T cells do the same thing. Here's E. coli, and I will never forget you. So sweet and loving, but really he's going to just kill E. coli. So, I mean, not so sweet, but, you know, kind of fun. Anyways, at this point in the video and in your learning, you should be able to tell me what immunocompetence means, which immune cells become immunocompetent in bone marrow, and which ones become immunocompetent in the thymus. Where do naive lymphocytes encounter their first antigen? What is an antigen presenting cell? What are antibodies? How do B cells become activated? How does the primary and secondary response to a pathogen differ? And what are memory cells? I'm going to stop here, and we'll get our next video up and running just in a few minutes. Bye.